Good morning, everybody. Are you all well? Yes? Well, you'll wake up as we get going, no doubt. My name's Dr. Hilton, and I'm going to do the first session, which is going to be introducing you to what I believe is a wonderful world, as well as the invisible world of microbiology. But I just want to introduce you to my colleagues as well, who you're going to see over the 2 days. On the front here is Dr. Worthington, who's a clinical microbiologist, and he's going to talk to you about infectious diseases and how we can protect ourselves from infection. And we've also got Dr. Ashraft and Dr. Brown, who are going to help us with the practical aspects of today, because you're going to get a really rare opportunity to do some practical microbiology. And many people spend their entire careers and never even get a chance to grow bacteria and look at bacteria in the way that we're going to introduce them to you today. So I hope that you really enjoy it, and I hope that you'll get something out of it. And because I don't just want you to sit there and listen to me talking, I've given you these cards here, which you should all have got one on. And one side says yes, and he's green and happy, and the other side says no, and he's red and a little bit miserable. And I'll want to interact with you throughout the lecture, and I want you to show me this card as we go along. So if you think something's good or you think it's right, then show me the green side. Or if you think something's bad or wrong, then show me the red side, okay? That's what we'll do with these cards as we go along. And you've also got these books here that we've given to you, the book that accompanies the course, and each section is a different colour. And just in this first session, we're going to go through the green section, which is just to introduce you to some microbiology, because I'm conscious that you might never have even studied microbiology before. So let's have a little go with these cards then, and maybe you can show me. Have you ever studied any microorganisms, any bacteria in your science classes so far? A few of you have talked about them with your teachers and things like that. Okay. And what about, say, for example, we're all microbiologists. Do you know what we do? Do you know what the job of a microbiologist is? Yeah, a few of you do. Okay. And can anybody tell me, if you watch the news or your parents watch the news and they say things to you, can you tell me any bacteria or viruses that are currently in the news at the moment? What about you, young man? Swine flu, things like that. That's right. So microbiologists, we deal with bacteria, which is what I'm going to talk to you about now, but we also look at fungi as well, and we look at viruses and other parasites and protozoans and things. But today we're just going to talk about bacteria, which hopefully you've heard of. So I want you to use your cards with me now for a minute. This spiral that I've got here is the history of the world on one slide, okay? And it starts here 4.5 billion years ago, okay? Incredibly long time. And as it starts to spiral round, we're getting nearer and nearer to current time as we go around the spiral all the way around here, and here is today, okay? So what I want you to do is, as I move the laser pointer up the spiral, I want you to raise your card when you think we believe bacteria first populated the Earth, okay? So how long do you think they've been here? So this is the beginning of the Earth, 4.5 billion years ago. So some of you think they were here right at the very beginning. And as we come along here, this is 3 billion years, okay, coming around 2 billion years ago, 1 billion years ago. Still some of you not sure. We're coming around here now. We're coming up to 250 million years ago. Still you don't think they were here? 6 million, 5 million, 4 million, 3 million, 2 million, 2 million years ago, 1 million, 100,000 years ago, yesterday. I'm slightly concerned that some of you still haven't put your card up. So at some point, I assume between the beginning of time and where we are now, bacteria originated on the Earth. And to our best estimates, that was probably somewhere around maybe 2 to 3 billion years ago. So they're actually one of the first inhabitants of Earth, okay? Now let me ask you another question then. How long ago do you think we actually discovered bacteria? How long ago do you think we actually knew they were here? Although they were here 3 billion years ago, if we start, say, here, 250 million years ago, and I come round here, raise your card when you think we knew about them. So we come up to 6 million, 5 million, 4 million, 4 million some of you, 3 million, 2 million, 1 million, 100,000, coming up to 100 years ago maybe, a few more, and then today. Well, I saw quite a few cards over here go up when we sort of got to about 100 years ago. It's actually about 300 years ago that we've known about microorganisms. And we know about them because of the work of a guy called Anton von Leeuwenhoek. And this is him here. And what he actually did is he took a toothpick and he picked about in his mouth, And about 300 years ago, he looked down these very crude microscopes that he's made. They weren't really like microscopes, not like the one I brought here, but they look more like magnifying glasses, really. But they had the power of magnification of a couple of 100 times, maybe 200 to 300 times. And he drew in some pictures in a special papers that he wrote to other scientists. He drew these here. And the more we go through this course, you'll see that this was probably the first time that he described certain bacterial cells that look like footballs, which we'll come to talk about in a minute, and certain bacterial cells that were long, almost like baked bean type structures, long rod-shaped cells. So we very much attribute the discovery and first description of microorganisms about 300 years ago to a guy called Anton von Leeuwenhoek. But you'll see that at the time, when he tried to explain bacteria to people, they weren't really very interested, because scientists were considered to be a bit weird and wacky. Put your cards up if you think scientists are weird and wacky. Oh, see. Well, we are. It's absolutely true. So people view science with a little bit of skepticism today, but you can imagine 300 years ago that a scientist saying that discovered this invisible world that nobody else could see, everybody thought he was a little bit mad, really. And so there's a very big gap between the 1600s and the work of Louis Pasteur in 1857. Now again, put your cards up. Who had cereals, for example, for your breakfast this morning? And did you have milk on them? Yeah? Some of you did? No? And the milk that you had on there would have probably been pasteurized, and that's named after Pasteur, who was a French chemist and scientist who actually discovered an awful lot about microbiology, and his work is commemorated in this note here, which is a 5-franc note. Now, France is currently using the euro, isn't it? But before they started to adopt the European currency, they used francs. And they actually commemorated the work of Pasteur on the back of one of their notes. And we can see here, there's a young guy here called Joseph Meister, who's actually fending off a dog that had rabies. And at the time, rabies was incurable, and many people died from it because the animals like dogs and foxes and things carried rabies, and if you got bitten by a dog, you'd get rabies and die. Now, Pasteur actually worked on a vaccine, and this was the first time that he'd used this vaccine, and he managed to save Joseph Meister's life. So the first thing that he's attributed with is actually developing a vaccine against rabies, which is very important. As we go around here, you'll see there's some sheep in the top left-hand corner, and sheep were susceptible to anthrax. And again, Pasteur developed a vaccine for anthrax. You can see there's some chickens in the right-hand side here. He developed a vaccine for cholera in chickens. He also developed vaccines to protect the silk industry at the time, and all these apparatus in the middle here he used for a very important but very simple experiment, which was to disprove the spontaneous generation theory at the time. And that was that people thought that things just happened. They thought that grains of rice and grains of wheat just turned into rats. That was what happened. It just happened. They didn't want to question it. They didn't even care how it happened. All they knew is it just happened. And that was what they call spontaneous generation, that you might just suddenly turn into a dog or suddenly turn into a pig, something like that. It seems absolutely ridiculous that they would think that, but at the time that's what they thought. And they believed that food just spoiled, or that wine just turned into vinegar, and milk just turned into cheese. But what was actually happening was that microorganisms that we now know are invisible to the naked eye were actually causing food to spoil. They cause milk to eventually turn into cheese. They cause lots of other things that we can't see, and he proved that with these experiments. So Louis Pasteur was a very important experiments he did at the time. The final person I want to introduce you to is a sort of pioneering microbiologist, is this gentleman here called Robert Koch. And he was a guy that actually proved the germ theory. Have you heard of germs? Stick your hand up if you've heard of germs. Yeah, you have? Nearly all of you have heard about germs. Well, germs, as we perceive them, are if I was ill and I coughed, then those germs would go from one person to another person and they would become ill. So it's the fact that bacteria and viruses can be spread and spread disease. And this gentleman here was important, a medical microbiologist, in showing that bacteria can spread disease from one place to another. And his wife, incidentally, was also very important in actually developing these solid culture media, which you'll be using later to grow bacteria on. His wife was actually a very good cook, and she loved making jam at the time. And all the early microbiologists we didn't really know what to grow bacteria on. And it was actually Robert Koch's wife that helped him design agar plates, which is now one of the main bits of equipment that all microbiologists use in hospitals, in industry, and later we'll introduce you to them so you can see how they work. A few years ago, I asked some children, what do bacteria look like? What do you think bacteria are? And these were some of the best pictures that were drawn for me. And the honest answer is that you don't really know, because you've never really seen them, but you've got an idea of what they probably look like, and this in the sort of a young student's mind's eye is what they think bacteria look like. In reality, I could say that they look like this, and you might think, nah, they couldn't ever look like this really. They don't have eyes for a start, but the thing is that we do have is that there's some of the basic structures that we can see are represented in these little diagrams. So many bacteria are round, almost football-shaped type cells, and I'll show you some of those later. And the other major group are those that look like baked beans, very rod, we call them rod-shaped bacteria. But there are other specialist bacteria, some of them are curved and a bit spiral-shaped, and many of them have these sort of things hanging off them, these appendages, which either allow them to stick onto things or allow them to swim and allow them to move. So there are certain features that we can pick up from these cartoons about what bacteria really look like, but I will show you some proper bacteria in a minute. If we look at a cartoon drawn of how a bacteria actually exists, you can see that it's very simple, okay? They are very simple cells. Have you studied human cells or plant cells, for example, in your science classes? A number of you have. So you'll know that human cells are actually quite complicated. And one of the main things that they have in the cell is a very defined nucleus, okay? Who can tell me what's inside the nucleus? Anybody? DNA, that's right. DNA is contained within the nucleus of your cells, of your pet dog cells, and of plant cells and things like that. But what you'll see in a bacterial cell is that the DNA is not contained in a nucleus. It's almost like a ball of wool just randomly scattered in the cell. And that is one of the fundamental differences between bacteria and human cells, is that they are much more simple in the way that they're designed, and one of the primary reasons is that they don't have a nucleus. They have DNA just randomly scattered in the middle. You then look around the rest of the cell, and there's a phrase that you might have come across called organelles, okay? And organelles are specialist bits and pieces inside human cells that allow us to do all the wonderful things that our bodies can do. Bacteria don't have those things. They are very simple. They have no other components in there. They have some rather specialized cell walls to keep everything together. They have some DNA in the middle. They have a few bits and pieces to make proteins and things like that, just like us. And sometimes they can have these external appendages, for in case of this one is called a flagella, and that allows the bacteria to swim. So bacteria, when we look at them in a sort of generic kind of template way, they're simple, they have no nucleus, they don't have any specialized bits and pieces, but often they can have some structures that allow them to swim. So let's ask ourselves a question then. Well, why can't we see them? Why can't we see bacteria, and therefore why do we not know that they're there? And the honest answer and the simple answer is because they are very, very, very small. And a typical rod-shaped bacterium like this, one of our baked bean type of bacterium, would be about 2 micrometers long. And a typical football-shaped bacterium like this would be about 1 micrometer long, okay? So if you put your hands in front of you like this is about a meter, okay? It's about a meter rule. I'm sure you're all familiar with that, yeah? And as you bring your fingers together, to about the width of one finger is probably about a centimeter, okay? Now if you imagine on your ruler, the finest division you can see in a normal ruler is a millimeter, okay? If you imagine now dividing that up 1,000 times, that would be a micrometer, okay? So there are 1,000 micrometers in a millimeter, okay? Which is the smallest division on your ruler, which is why you can't see them, because they're so incredibly small. But let me focus you in a little bit more. Hold your hand up if you think you know what this is. This is something I put under a very powerful microscope in our laboratory called a scanning electron microscope. And if you think you know what that is, put your card up. Yes, young man, what do you think it is? Lead. Is it lead? That's a very good guess, but it's not right. Anybody else want to have a go? Anybody else think that they know what it is? What about I give you a clue? Here's a clue for you. What do you think this is? It's a pin, that's right. So this bit here, this is actually the tip of this pin here, okay? And before I put it under the microscope, I actually took some bacteria that were swimming around in some liquid, and I dipped the end of the pin into the liquid, okay? And then I put the pin into the electron microscope. And then I started zooming in. So when we zoom in, can you see what's happened now? That the top of the pin, this is the top of the pin, and it looks a bit like a mountain really under this magnification. You can see how flat the tip of the pin is really, and you can just about see some of the molecular structure of the stainless steel on the side. And if you look very carefully, you can see something on the side. So let's zoom in again, shall we? So we're zooming in again, and now you can see there are some individual cells here, these little rod-shaped cells on the side. And if we zoom in again, this is zoomed in probably about 1 million times now, you can see here these individual cells of a bacterium called E. coli. And E. coli is in your gut. In fact, there are more bacteria in your gut right now than there are human cells in your body, okay? So although you don't think you know E. coli certainly knows about you. So, we're talking now about a minute scale of biology, okay? We're talking about microbiology. And we're talking about micrometers, okay? And I've tried to focus you down into this world where things are incredibly, incredibly small. But let me give you another reference to make you realize just how small this is. If you were to put a Smartie, okay, in the middle of your hand, and if a bacterium was to grow to be as big as that Smartie, how big do you think your hand would have to grow, okay? And I want you to think about it in terms of football pitches. So think about if a bacterium grew on your hand to be as big as a Smartie, how big would your hand have to grow in order to keep everything relative size? And I'm going to give you a few seconds to talk amongst yourselves and discuss what you think might be the answer. Okay, so as big as a Smartie, how many football pitches would your hand have to be? How many do you think? You think Old Trafford and Villa, so you're thinking 2? Yeah. Would it have to be Villa, would it? Are you a Villa supporter? West Brom. So it could be Man U, West Brom and Villa then. Okay, everybody, so how many do we think? We've got 3 down at the front here. He thinks it will be West Brom, Man United and Villa. Is that right? Yeah? Anybody? I think it's like 2 Wales. 2 Wales? Yeah, but in terms of football pitches? 4 football pitches. 2 football pitches. Any more than that? 7. We're getting closer. Any more than that? 9. He's got it. 9 football pitches. That's exactly right. If you imagine your hand being stretched out to the size of 9 football pitches, then any bacteria on your hands, and there are lots on there, and we'll show you them later, would be about the size of Smarties. So I hope that gives you some idea of just how incredibly small bacteria are. Now, so far, we've been talking very sort of hypothetically, haven't we? We've been talking about pictures and diagrams and what things look like under electron microscopes. But I wondered who would like to help me see some real bacteria right here and now. Anybody would like to do that? Yeah? Anybody? Who would like to do it the most? Yeah? Anybody? No? I'll tell you what, you answered the question right, so you could come and help me if you like. How about that? Okay. Come up with me then, please. What's your name, young man? Mohammed? Right, Ahmed, right. Who's here to help me now. Now, do you remember I said that 300 years ago, Anton von Leeuwenhoek, okay, who was a gifted biologist, but he didn't know anything about microorganisms, what he did is he took a toothpick like this. So I'm going to give you that toothpick like that, okay? And what he did is he picked about in his mouth. So I want you to pick about in between your teeth and get me some plaque on the end of that. That's all you've got to do. That's all he did. Okay, have a good dig around, come on. Get those Cocoa Pops out or whatever. Oh, I'm going to need more than that. Go on, have a good dig around. Your braces will help as well. Go on, there's some goo on there, I need some. Let's have a look. Yeah, you keep hold of it, that's fine. Now then, what I've got here is a microscope slide, okay? So it's a very thin piece of glass, okay, which will allow the light to come from this microscope. The light will come up through here, through the preparation that we're going to put on here, through these series of lenses, and I'll be able to see what is on here. But first of all, we need to make the preparation. So come around here, please. That's it. So there's your glass slide. And what we've got here is a pipette, okay? And this pipette is actually called a Pasteur pipette. It's named after Pasteur, who we've already had a quick talk about. And in here is just some water, okay? So what I want you to do is to take this pipette, take the water there, dip it in, suck up some water, and I want you to put me 1 drop of water on that microscope slide, okay? That's all you've got to do. You can rest that down for a minute. That's it. Go on. Put some water on there. Go on, I'll keep talking while you do that. Tell me when you finish, yeah? Is that all right? Yeah, good, good. Okay, so what he's going to do, he's going to put a bit of water, excuse me, he's going to put a bit of water on the microscope slide, and then yeah, that's right, yeah, take it, just squeeze it and get some water up, like that. That's it, and then just put a drop on there. 1 drop, just squeeze it gently. On there, on there. That's brilliant, that's good. So he's put a little bit of water on there. And now what we're going to do is to take, that's it, you can just rest those there, that's fine. So can you remember which end of the stick it was? It was that one there, wasn't it? So I want you to get those, and any microorganisms which you can't see, if you could see on the end of this stick, there's nothing there, I want you to rub that in the water there, okay? As though you're kind of colouring it in, so to speak. So that's it, put it out. So any bacteria that he's got out the plaque out of his mouth, he's pulling out of there, and he's now going to put them, and they're going to be swimming, okay, in the water at the moment, okay? And in order to see them, that's it. In order to see them, we've got one last thing to do. Now, you probably can't even see this, but I'll hold it up. Can you see that? That's called a cover slip, okay? It's a really, really thin piece of glass which we're going to trap the liquid, the water, with the bacteria in between this piece of glass. So what I want you to do is to put that piece of glass on top of the liquid. You can put that down now, that's fine. Put that on top of there for me, please. So make a sandwich. Wonderful. That's brilliant. Thank you. You can go and sit down now. Thank you. Give them a round of applause, everybody, please. Okay, so this is what I've got, which you can see. I've got a microscope slide. I've got some water with some tooth scrapings on like this and a cover slip. And I'm now going to have a look at this under the microscope, and I'll show you just how many bacteria are in your mouth, okay? Okay. Right, now you want to be able to see what I can see, don't you? So what we've got here is a really sophisticated camera which is actually looking down the microscope, and it's coming down here, and I can get a feed over to my computer, and I can show you on the screen what we've actually recovered from the mouth there. So let me see if I can get this up on the screen for you. Here we are. Can you see that? Look at that. Isn't that amazing? So can you see that there are bacteria swimming around? Can you see that? This is a big, called a fusiform. That's called a fusiform. That's a very big bacterium. But you can see they're all moving. They're all swimming. And do you remember that I showed you the picture, and some of them had this little tail, this little flagella at the back. They're busily spinning that round. Okay, everybody, I need you to listen to me, okay? They're busily spinning that round, and that is what is making them move, okay? So they're swimming around. And this was what Anton van Leeuwenhoek saw over 300 years ago, where the first description of bacteria was almost the same as the experiment that we've just done now, okay? He didn't see pictures as well as this, because this microscope can magnify about 1,000 times, and his microscopes weren't as powerful as that. So he wouldn't have seen them quite as large as this, but this is what he saw that inspired him to write about microbiology. Now, what is happening with these organisms is that they are busily feeding on, can you see there's some material here? These are probably bits of food or breakfast or whatever that have been left there, and they're busily eating that. That's what they want to do. And when they eat that food, they produce acid. So they eat the sugar and they produce acid. And who knows what acid might do to your teeth? Any idea? Exactly, that's right. And that's why your teeth eventually fall out if you don't clean them. But let me tell you now that although you may be worrying about kissing this young man in the future, if we'd have done any of your mouths, it would have been exactly the same. And if I'd have given you all a toothbrush and some toothpaste and sent you away scrubbing for the last 20 minutes and you came back, I would have still seen this, because you can just not get rid of these microorganisms out of your mouth. They're so intimately associated with all the nooks and crevices and under your gums and things like that, you can't get rid of them. So what you have to do, it's a daily battle, isn't it? Every morning and every evening, you clean your teeth, you clean your teeth, you clean your teeth. You'll never get rid of them. It's just to keep the levels down so they don't produce too much acid to make your teeth fall out. So, to thank you very much for helping me with that experiment, there you go. There's a salmonella for you, which you can have. Thank you. Now, what we've established is that we've looked at bacteria, we've determined how small they are, we've looked at some of the features that they have and some of the properties, and you can learn more about these in your handbook because there's lots of more information in here. All the slides that I've shown you, you can see are in there, so you can read about them in your own time to learn a little bit more about them. But what I want to get across to you is that it's very easy to think that all bacteria are bad. And indeed, several of them are bad, and it's those that we need to protect ourselves from. And we're going to teach you about that later today. Dr. Worthington will talk about infection control, and later we'll talk about food safety and also sexual health safety and sexually transmitted diseases, all of which can be spread by bad bacteria. So let's play a little game now, shall we? We'll play the bad-good game. And I first want you to think about salmonella, okay? And I want you to show me your card. I want you to show me the green side if you think it's good, and I want you to show me the red side if you think it's bad, okay? So vote now. Lots of reds over there, a few greens, mainly red, okay? So let's see what it is. Oh, it's not, it's bad, okay? So salmonella is an organism that we'll talk about more tomorrow. But salmonella is one of the major causes of food poisoning, of foodborne infection, and it's typically associated with poultry and eggs and things like that. And tomorrow, I'll teach you how to protect yourself from bad bacteria such as salmonella, okay? What about this one? Lactobacillus. What do we think about that? Have you heard of it? Good or bad? What do you think? Let's see your cards. Lots of greens this time, a few reds, but mainly green. Okay, let's see if you're right or not. Actually, it's a good bacterium. Lactobacillus is an organism that we use in the food industry, for example, to help us make yogurt and to help us ferment foods like salamis and things like that. But it's also used a lot in these types of drinks. I don't know if your parents buy these kind of Danone, Yakult, and these other types of probiotic drinks. They all contain lactobacillus. It's very good at producing lactic acid, and lactic acid is good at keeping your gut nice and healthy. What about Campylobacter? What do we think about that one? Got lots of red actually. Okay, let's see then, shall we? You're right, it's bad. It's bad. Campylobacter is actually the #1 cause of bacterial food poisoning, and many people haven't heard about it. So I'm very impressed that you actually know that Campylobacter is bad. And it's again associated with poultry, and it's associated with undercooked chickens and things like that. So I'll teach you tomorrow about how to protect yourself from Campylobacter. What about the next one? Staphylococcus. What do you think about that? Good or bad? We've got some greens, we've got some reds. We've actually got a complete mixture with this. So let's see what this one is. Ah, see? So there you go, you see, you're either all right or all wrong, I assume. Staphylococcus is an organism that lives on your hands. Okay, listen to me. I'm trying to explain things to you, so I need you to listen to me. Staphylococcus is an organism that lives on your hands, and later, when we go into the laboratories, we'll probably grow Staphylococcus because we'll be sampling your hands. So the Staphylococcus aureus and Staphylococcus epidermidis are two species that we undoubtedly see, and you might have heard of Staph aureus, and Staph epidermidis lives on the hands where it causes no problems at all, really, and actually it's quite healthy to have microorganisms on your hand. It keeps your hands in good condition, but we'll see later that the hands can be very important vectors of disease, but in largely there are bacteria on your hands that cause no problem at all. But who's heard of MRSA? Have any of you heard of MRSA? Lots of people over here. Yes, MRSA. That stands for methicillin-resistant Staph aureus, and I'm sure you know that that's bad, okay? And it's an example of a microorganism that can occur in people who, for example, have had surgery and the skin has been cut and the wound becomes infected with an organism that's very difficult to treat with antibiotics. And Dr. Worthington will talk more about MRSA later when he talks about superbugs, okay? So MRSA is an example of a superbug, and that is very bad. So what I want you to think about is that although some microorganisms are obviously bad and some microorganisms are obviously beneficial, there are some microorganisms that can, under the wrong circumstances, actually cause us harm. And it's those that we need to be mindful of, because it's those that we need to protect ourselves from, particularly the superbugs. One last one then, chlamydia. Who's heard of chlamydia? Yeah? Is it good or bad? Lots of bads over here. Bads over there, bads over there. Let's have a look then. Chlamydia is bad. That's it, correct. That's right. So chlamydia, the bacterium chlamydia can cause a sexually transmitted disease, which Dr. Worthington will talk about as part of many other bacteria that can cause disease. Okay, everybody listen. I can't speak over you. If you're talking, then I can't talk. Okay, so if you listen, just another 5 or 6 minutes just to listen, and then we'll do something else. So just bear with me a little bit longer. But very importantly, chlamydia is a sexually transmitted infection that in many circumstances people don't know they have, but it can actually cause quite serious implications later in life if you want to have a family or something like that, for example. And so if you don't know you've got the infection, you can be spreading it around to other people. And Dr. Worthington will talk more about chlamydia and how you can protect yourself from chlamydia. Okay? I just want to finish this introductory session by describing one of the parameters or one of the abilities of microorganisms that makes them such a big problem for us, and that is the speed with which they can grow. Okay? If, for example, you've got a little brother or sister, or you've got plants, or you've got a dog, you've probably watched it grow up over years and years and years, and it's got bigger and bigger and older, but it's taken a long time, okay? Bacteria are completely different. Bacteria can grow incredibly quickly. They can go from one bacterium to two bacteria in a matter of maybe 20 minutes. And then within another 20 minutes, those two can have divided into 4. And then another 20 minutes, those 4 can have divided into 8. So this is a situation where things can very quickly get out of control. And if we think of the circumstance of like food, for example, if you allow bacteria to grow on food, even if only for a few hours, the organisms can quickly grow and cause toxins and actually make you ill if you were to eat them. So it's important that we understand that bacteria are able to grow very quickly and that we have to be mindful about what allows them to grow. So on the next slide, I'm going to show you a little video of bacteria growing under a microscope. So what we did is we put a single bacterium, just one cell, under the microscope, and we filmed it on the microscope as it divided into 2, and as those 2 went into 4, 8, 16, 32, and so on. I want you to watch how quickly it grows, okay? Here we go. So you see that? So how quickly that happens. So every 20 minutes it's dividing. So if you left some food out overnight, for example, you forgot to put it in the fridge when you went to bed, for example, the next morning you thought, oh, I'll eat that, then you could be eating a whole load of bacteria that could make you ill. So what actually stops bacteria from growing? Well, one of the best things to prevent the growth of microorganisms, particularly an environmental factor that we can all easily look at, is temperature, okay? Bacteria are entirely really susceptible to temperature, and we see that they have this type of response that looks a little bit like this. That for many of the bacteria that cause disease in humans, which we're talking primarily about in the next 2 days, most of them are unable to grow very well above 50 degrees. Many of them are unable to grow very well below 10 degrees, and they have an optimum here somewhere in the middle. What do you think this optimum temperature might be? 35? 37, that's very good. And what's the significance of that? Body temperature, that's exactly right. So many bacteria that are good at causing disease in human grow very well at body temperature, but they can be inhibited by keeping things cold, in the fridge for example, and they can be killed by taking the temperature above 50, as in proper cooking. And that's something that I'm going to concentrate on with you tomorrow, about how we kill bacteria with cooking and how we can stop bacteria from growing by keeping things properly in a refrigerator. The final thing that I want to say is that bacteria can spread very easily, and that's why we all need to be responsible, not only for ourselves, but for each other as well. Because, for example, if I went to the toilet and didn't wash my hands, okay, and then I came over and I shook your hands and said, hello, pleased to meet you, how do you do? And then you go and eat a sandwich, he's going to be ill, and it's my fault, isn't it? So I need to look after myself, but I also need to be mindful about it for everybody else, because bacteria can spread by unwashed hands, they can spread by sexual contact, and they can spread from inanimate objects, things that we might touch, like pens or chopping boards or door handles and things like that. So we always need to be mindful about where bacteria are and how we can protect ourselves from them. And over the rest of this afternoon and tomorrow, we'll be looking at superbugs and infection control, and Dr. Worthington will lead you through that. And we're also going to go in the laboratory and do some experiments on hand washing and looking at what bacteria are on your hands. But we're also going to add an extra little bit as well this time. We're going to allow you to sample some other bits and pieces. So if there's anything that you really wanted to know, does it have bacteria on, like your mobile phone or your pencil case or your lunch box or whatever you've got with you, then there could be an opportunity in your groups to actually sample some things, and we'll see what's growing on it for you. So it's such a wonderful opportunity for you to get something out of this. Okay, if you're excited and interested about microbiology, then the next 2 days are going to be really good for you. Okay? So thank you very much for your attention. Have you got any questions you'd like to ask me before we break into groups? Is that right, Dr. Brown? No? Is it break, is it? I'm sorry, before we go for break. We've got about 5 minutes, is that right? Yeah? Yes? Can we do it again? What, do you want to do it? You want to look in your mouth, do you? Okay. Well, we won't do it now because everybody will get bored watching you, but yeah, I'll do it with you. Yeah? No problem. Any other questions anybody you might have about microorganisms? Yeah? What part of you... Okay. I'll tell you what. Go on, speak into the microphone. Because I think everybody wants to hear your question. Go on. Sing a song.
What part of your body's got the least bacteria?
The least bacteria. Okay, thank you very much. Okay, everybody. We're talking about bacteria and we're talking about the body and where microorganisms might be. And the question I was asked is, where are the least microorganisms? Now, for your body, and the exterior part of your body is absolutely full of microorganisms. And it's particularly full of microorganisms where your body is kind of moist, really. So under your armpits, in your groin, in your mouth, in your intestine and things like that are absolutely full of microorganisms, okay? But yet there are some parts of your body where there aren't any microorganisms. So for example, within the meat muscle of your body, there shouldn't be any microorganisms. And within your brain and within your blood and things like that, there shouldn't be any microorganisms, because those are sterile places. For a long time, we thought that the gut was sterile, as in the stomach was sterile, but it isn't. There are certain microorganisms that can even live in the very low pH of the gut. So microorganisms of the body tend to be on the external surfaces and in the intestine in particular, in the urinary tract and things like that. Once they start to get into your muscles, into your blood system, into your brain and things, into your lungs, then you're ill, then you're unwell, and that's where there shouldn't be any microorganisms. Okay? Very good question. Would you like to ask a question?
Do you know what DNA means?
I do know what DNA means. Do you know what DNA means? Does anybody want to tell him what DNA means? Deoxyribose nucleic acid. That's what it stands for. Deoxyribose nucleic acid. Okay? It's the building blocks of DNA that allow us to replicate our DNA. And bacteria have the same DNA as us as well. Any other questions anybody would like to ask? Another one over here? Speak in there so everybody can hear. Go on. Sing us a song.
You know the part of the bacteria that makes it resistant to antibiotics?
Yes.
Can't you just cut that off?
Again, it's a very interesting question. I've been asked about resistance to antibiotics in bacteria and the DNA that encodes for that, and I've been asked, can we not just get rid of it? Can we not just cut out the DNA that encodes for that resistance? And the answer is it's a very good idea, and if you could actually do it, I suppose it would work, because a lot of the resistance that we see in bacteria is encoded within the DNA, and that allows them to spread it on to other people. But the actual practicalities of taking a microorganism and cutting the DNA out, in an infection there may be 1 billion cells, and you'd have to do it for every single one of them. So it's not really practically possible, but it is a good idea, and if it could be done, then that would be a strategy of doing it. Another one here?
How was swine flu formed?
How was swine flu formed? Okay. Well, swine flu is not unlike other types of flu in that it has two specific components, which we call the H component and the N component. And for those that you want to know, it's a hemagglutinin and a neuraminidase. That's what the two are called. H. Have you heard of H1N5 and things like that? They've got these different components. And what happens is when a flu virus infects somebody, all its genome falls apart, it replicates itself, and then it puts it all back together again. And the way that flu viruses evolve is that they actually infect, for example, a pig or an avian flu, a bird or something like that. And when the pig or the bird was sick with flu itself, and the human flu was there as well, it all recombines so that the product that comes out the other side is actually a mixture of the two flu viruses. And that's why you might get H1N4, H1N5, N2H4, all these different combinations. So it's a mixing together of viral infections when somebody is infected with two at the same time. That's how they evolve. Do you want to say the...
What's MRSA?
What is MRSA? MRSA is a microorganism. It's a Staphylococcus aureus organism, which is one that we typically find on the hands in about 40% of people. But the problem with MRSA is that it's learned over time to become resistant to many antibiotics, but importantly one called methicillin. And methicillin is an important antibiotic that's used an awful lot in hospitals, and because it's become resistant to that as well, doctors have very few options left to actually treat. So if you end up getting a wound infected with MRSA, it's very difficult to heal, and the doctors have very limited antibiotics they can use. And there are a few that they can use, such as vancomycin, but again, we're starting to get worried that microorganisms can become resistant to that as well. And if they do, then we're going to have very few options left. So MRSA is a very resistant microorganism, predominantly found in hospitals, but can be found elsewhere as well, that can cause infection of wounds in particular. Anything else? Yes? Do bacteria cause cancer? That's, again, a brilliant question, actually. There are some examples of where bacteria can cause cancer. There's a very clear one, which is called Helicobacter pylori. And this is an organism that lives in the gut in people that are infected with it. It can cause gastric cancer, and it can cause ulceration and things like that. So there's a very clear link there with bacteria and viruses. Another very strong link is with viruses and cancer, particular with the papillomavirus and cervical cancer. So again, viruses almost certainly, bacteria again almost certainly can cause cancer. Yes. Any other questions? Are we good? Lovely. Thank you very much.
Okay, well done.